Hi everyone, my name is Nicole and my presentation is on the paper Brain Imaging Reveals Neuronal Circuitry Underlying the Crow's Perception of Human Faces by Marzluff et al. 2012. The reason I chose this paper was because I've heard before that crows can somewhat recognize humans, but I wasn't really sure how true that was. Then while I was researching papers for this project, I stumbled across this study and I thought it would be really interesting to present. Okay, so these are a few key terms I figured you guys would like to know because I'll be mentioning them later on throughout the presentation. FTG is F18-fluorodeoxyglucose, and this will be administered to the test subjects before brain imaging. PET is positron emission tomography, which is commonly referred to as a PET scan. This will be used to scan the crows. TNA is nucleus tenea of the amygdala. The amygdala is the part of the brain that processes emotions, which you probably already know, but I figured I would just mention that. Okay, so a wide range of species like sheep and non-human primates have the ability to recognize human faces. This ability is linked to neural integration of perception, emotion, and memory. The network of brain regions that perceive and analyze faces utilizes the ventral and dorsal visual pathways. The ventral pathway allows for specific discrimination of something, and the dorsal pathway provides a faster but rougher depiction of something. So these pathways kind of work together at the same time. I mentioned sheep and non-human primates earlier because previous studies show that there are neurons in the brains of these animals that respond to human facial information. In a previous study, the researchers demonstrated that American crows do discriminate among humans based on their facial features, but there isn't any evidence of what happens on the neural level when crows exhibit this behavior. The point of the study is to investigate what actually happens on a neural level in these crows, and the hypothesis is that crows recognizing humans might involve a distributed set of interactive brain regions. The hypothesis is based on the reasoning that birds and mammals have the same common sensory and motor circuits. Crows were captured from big roosting and foraging groups on 3 separate occasions. It was ensured that the birds were not previously captured. Researchers used a net launcher to capture 30 large adult birds that were assumed to be males. The picture on the upper right is a net launcher. It works like a cannon and shoots a net out. This technique is used a lot to capture birds in a safe manner. The people who captured the crows wore threatening face masks. It's important to note that the Institutional Animal Care and Use Committee protocol was followed very closely. Crows were housed in individual cages for 4 weeks. As the crows were being looked after during this period, they learned the caring face. This was the mask worn by those feeding the crows and cleaning the cages. The masks were worn at all times. The threatening masks were worn while capturing the crows as well as moving the crows from the cages to the PET laboratory. I like to point out that all masks were of the actual people's neutral expressions, so relation was depicted by behavior, not of facial expression. So basically, if I was involved in this study, then my blank expression would be photographed and then used to create my own mask to wear. There is a figure on the next slide of the masks which will give you a better understanding of what they looked like. Okay, the night before imaging, the test subject was moved to a covered cage in the imaging lab to acclimate overnight. In the morning, the test subject was blindfolded, removed from the cage, administered 1 millicurie of FDG via an IP injection, which is an injection of a substance into the body cavity of an animal, returned to the cage for a 2-minute rest, and then shown the researchers wearing the threatening masks in 1-minute on-off blocks for 14 minutes. After the activation protocol, blindfolded crows were administered 3% isoflurane and oxygen with a flow rate of 300 to 800 milliliters per minute, acting as an anesthetic, and then imaged. Activation and image timing were based on data from dynamic imaging of a crow, which showed faster brain FDG uptake and washout than seen in mammals. Each test subject was assigned a treatment at random and scanned 1 time. This is a figure from the paper that illustrates the experimental steps. In the number 2 picture, you can see that the mask is of that individual person's actual face. It's easier to tell that this is a mask by looking at his eyes and seeing the circles around them, which depicts the eye holes of the mask. In the number 6 picture, it shows the caring masks and threatening masks, which basically look the same. But I just want to reiterate that the crows know the difference between these masks based on the behaviors that the people exhibited while in the crow's presence. Also, I didn't mention this before, but all crows were given proper care between 1 and 14 days in length in order to prepare them for being released back into their habitat. Alright, so now we're going to move on to the results section. So, results suggest that American crows recognize familiar human faces by evaluating visual sensory information in the context of learned associations. Figure 2 depicts 2 pet FDG images. The upper image is of a crow that is awake during uptake, and the lower image is of a crow under anesthesia during uptake. So the pattern of FDG uptake during visual stimulation showed activation of the crow's tectofugal visual pathway and a bunch of other forebrain regions. This activation model concentrated neural activity on the central fovea of each retina, and this stimulated a strong response by the nucleus rotundus as well as its target, the entopallium. In birds, this visual network resolves distant, complex, and novel objects, and it's important to visual discrimination tasks, pattern recognition, concept formation that allows for individuals to be recognized, and depth perception, which are all tasks relevant to the crows in this study. The lower image shows a similar regional distribution of tracer was not obtained in the crow brain that was under anesthesia during the uptake period. Figure 3 shows the differences in brain activation patterns of crows shown familiar human faces versus no human face. In image A, the brain on the left is of a crow shown a familiar face, and the brain in the middle is of a crow shown no face. The reddish-orange color depicts activation regions in the brain upon either seeing a face or not. So, the sight of a familiar human face, whether it be threatening or caring, activated the rostral forebrain, including the hyperpallium, symbolized by an H in the image, as well as a large region in the nidopallium, mesopallium, symbolized by an N and an M. In the middle image, the lateral striatum, symbolized by LST, was activated. So the differential activation of the hyperpallium and the use of the tectofugal visual pathway means that crows use their thalamofugal pathway to perceive humans. The rostral nidopallium has strong connections to the entopallium and mesopallium and has access to the sensory information from the tectofugal and thalamofugal visual pathways. So this explains why the activation of the nidopallium is important to the recognition of human faces in the crow brain. The graph just gives you a visual of the differences in the activation pattern between crows shown a familiar face versus no face. The data for the crows shown a face is the dark circle, and the data for the crows shown no face is the light triangle. So you can see by the graph and the reddish-orange regions that there's a clear difference between the regions activated in the crow brains that were shown either a familiar face or no face at all. So we talked about how there's a difference in activation patterns between crows shown a familiar face versus no face. So now I'll be discussing the differences between being shown a threatening face versus a caring face. Different regions of the brain were activated dependent upon the face shown to the crows. Figure 4 is an image of brain activation patterns of the crows shown each mask. When the crows were shown the face of their captor, the brain activated the nidopallium, mesopallium, archopallium, including the region containing the nucleus tenae of the amygdala, and the nuclei in the dorsal thalamus and brainstem. These regions are shown as the reddish-orange areas in the first row. This response is elicited by the emotion of fear. When the crows were shown the face of their caretaker, the hyperpallium, mesopallium, preoptic area, and medial striatum were strongly activated. These areas are denoted as the reddish-orange areas in the middle line of brain scans. These regions are important to associative learning, motivation, and autonomic functions, including hunger in most vertebrates, suggesting that crows perceive the association established between the caretaker and food. It seems that in crows, as well as other animals including humans, the striatum and the amygdala are crucial to associative learning. The striatum is receptive to prediction areas, while the amygdala is receptive to the reliability of threatening cues. The results demonstrate how crows use a bunch of different regions from the brainstem to the forebrain to determine and adjust the response to individual human faces. This is actually consistent with current scientific views of how the subpaleolimbic network interacts with the integrative forebrain to shape memory-based social behavior in vertebrates, including humans. The use of cortical sensory processing, the striatum, and the limbic system suggests strong analogy and possible homology between mammalian and avian facial recognition and associative learning systems. So, in regard to future research, more studies are necessary to determine if the forebrain regions used by crows in this study are functionally analogous to facial recognition regions in humans and other mammals. It's important to continue the partnership between neuroscientists and ecologists, like in this study, because this will help researchers better understand the neural basis of cognition in wildly diverse animals. This knowledge can help researchers to generalize important findings across species and sensory modalities, develop strategies to reduce stress in captive animals, shape animal actions to decrease human-wildlife conflicts, and engage the public to be appreciative of the cognitive ability of other species. I'd also like to add in my own hypothesis I thought of as I studied this article. Since crows are able to recognize humans based on their behavior around them, then if the humans' faces were fully covered, would the crows still be able to recognize individual humans? Maybe this can be a potential future experiment one day. All right, here are my references. I really enjoyed creating this presentation and I hope you found it informative. Thanks for watching.